everyone. My name is Cassie McGinnis. I am a master's student at Illinois State University, and today I'm going to be talking about my favorite little vampire, Pedicularis canadensis, and the impact it has on its local tallgrass prairie community, specifically one that is invaded by Lespedeza cuneata. So the reason that I'm going to play the vampire is because it is a parasitic plant. So it uses these haustoria to connect to a host plant where it then takes carbon, nutrients, and water from that host back into itself. Specifically, though, I am going to be focusing on a hemiparasite. I already have this reaction. And so hemiparasites, they fill up after the host, but they also have functional chloroplasts and they're forming photosynthesis. So there are many species of hemiparasites, lots that can attach to roots, and that can be abundant in prairie ecosystems where they can help drive ecosystem processes. So if species differ in susceptibility or tolerance to parasitism, then a hemiparasite can in fact be present or abundance of these species, and therefore can alter a community. And hemiparasites can also decrease or reduce host growth and production and performance, as well as reducing community productivity for cutting out of above ground biomass. So this is a good introduction for, again, my favorite little vampire, Pedicularis canadensis, also known as Canadian wood betony. So this is a native root hemiparasite that's often found in open woodlands and prairies, and it can significantly affect prairie productivity and composition. This is also a plant that's of interest to different land managers as a means of potentially increasing diversity or as a sort of native biocontrol against undesired or invasive species. So hemiparasites such as pedicularis are postulated to alter biodiversity and community productivity through a few different pathways. For example, through direct parasitism, where they may act as a keystone species. Of course, I'm defining a keystone species here as a species that's relatively small in its number or biomass compared to the impact it's having on this community. And according to this hypothesis, pedicularis would increase biodiversity by suppressing competitively dominant species, thereby allowing competitively subordinate species to then become more abundant. However, hemiparasites could also influence their community more indirectly, in this case, more as an ecosystem engineer. So, here in this hypothesis, I would predict that pedicularis is able to alter nutrient availability through its nutrient-rich senescent leaves, which can also impact the nutrient processing soil microbial community. And overall, in grassland management, it is assumed that hemiparasites increase diversity by suppressing these competitively dominant species, allowing native subordinate species to become more abundant. However, this can also increase opportunities for invasive species to potentially get established and find foothold. And this leads into my nemesis on the prairie and my other focal species, Lespedeza cuneata, also known as silky bush bloater. So this is an invasive species native to Eastern Asia and Australia. It is a perennial legume. And while there are many different hypotheses for invasion success, I'm really going to be focusing on one for this talk, and that is the limiting similarity hypothesis. So the idea here is that an invader is more successful where functionally similar species are absent. So in this case, Lespedeza cuneata would be more successful in patches of the prairie where the native conjure Lespedeza capitata is not. So I'm also going to look and see like those pedicularis play a role in how cuneata is invading the prairie as well. So I know my research out of the giant bush prairie. This is a reconstructed tallgrass prairie, and there were 96 1-meter square plots that were created out of the prairie back in 2006. And past research at the site has shown that pedicularis does have strong facts where it is abundant, and that cuneata is it increases most where the native conjure Lespedeza capitata is absent. So I returned to this prairie to examine how these effects persist and how pedicularis is producing these effects on its community. So I did this by collecting soil samples from all the plots, where I then looked at the microbial carbon biomass from the soil samples using the microbiome protocol. And then I also sent portions of these soil samples out to Craftsman, where they use the Illinois soil nitrogen test to look at the amount of organic nitrogen within the soil. And then in each of the plots, I also did a community survey where I looked at the which species were present, identify them, as well as the percent cover or that abundance of each species. And then I put all of this together using a piecewise structural equation model, which is a form of path analysis. The idea behind these is they take a bunch of different variables, and they put links between them, and then you test causal connections between those variables. And the goal of this was to determine direction and strength of different pathways for Pedicularis is affecting its plant community, soil microbes, soil nutrients, and that invasion of cuneata. So I'm going to be showing you a few different models up here, and these are essentially all the variables that are going to appear in all of the models. So every single model is going to have the same variables present so that all the models are comparable to each other. A key note for all of these is that all of the data from these different variables is from 2024, except for the two variables that clarify them from 2019. And then all of the variables will be connected either by direct effects or correlations. And which variables are connected by those direct effects is going to be dependent on the hypothesis they're testing. So direct effects are going to be these solid arrows that point from the predictor variable to the response variable. Bold arrows are going to be significant effects, and they're going to be associated with a path coefficient in the initial models or the final, like full models I show you. And then double-headed dashed arrows are going to be the correlations. So here we assume that the variables are associated in some way, but we're not predicting a specific causal connection like we do with the direct effects. So when I'm looking at the models that are comparing the two hypotheses that focus on Pedicularis and Cassie community, so that keystone species of ecosystem engineer hypotheses, the model for the keystone species hypothesis did the best at describing the data. So starting to pick this apart, we see that 2019 Pedicularis cover has a positive direct effect on 2024 Pedicularis cover, and then 2019 cuneata cover has a positive direct effect on 2024 cuneata. So what this means is that where those two focal species were historically, they're more abundant 5 years later. Now focusing more on Pedicularis' effects, so we see that the historic Pedicularis has a negative direct effect on 2024 form cover, but a positive direct effect on 2024 legging cover. So again, wherever we see this historic Pedicularis, we see 5 years later that there's tends to be less forms, but more leggings. Now, when I'm looking for a keystone effect in this model, what I would expect to see is Pedicularis to be affecting one of the more competitively dominant species. So that would be either granuloid cover or 2024 cuneata cover. And then those dominant variables affecting the competitively subordinate species. So for legume or capitata cover. So really to get at the keystone effect, I'm not quite there with this model because I really need those indirect effects of how Pedicularis is affecting the forbs, legumes, and capitata through those more dominant variables. And so the indirect effects are a work in progress that I'm still actively trying to determine. So as of right now, while this model did better than the models for ecosystem engineer, I still need those indirect effects to see if it's actually acting as a keystone species. Or for the limiting similarity hypothesis, we again see that the two focal species in the past have positive direct effects on their 2024 covers. So wherever those groups were historically, they tend to be more abundant 5 years later. There is, I would say, support for the limiting similarity hypothesis because we see a negative direct effect from 2024 capitata to the invasive cuneata. So essentially, wherever there's more of that native Lespedeza capitata species, there is less Lespedeza cuneata. I will note that in this, we do not see any significant effect of Pedicularis on either of the Lespedeza species, which is unfortunate. We hope you have either. So, some takeaways from all of this, starting with the keystone species model. So, we do have these historic Pedicularis covered, leading to direct increases in 2024 legging cover, but decreases in form cover. And I do need those indirect effects to really know for certain if it's acting as a keystone species. However, through these direct effects and the potential for a keystone species effect, pedicularis may be useful to land managers as a means of maintaining or increasing or achieving diversity goals within their prairies. And then there is support, I would say, for the limiting similarity hypothesis because increasing cover of that native Lespedeza capitata did result in significantly less cover of the invasive Lespedeza cuneata. So this could be a help to land managers just to help them be more informed about make more informed decisions when dealing with Lespedeza cuneata and have a potential solution for maybe trying to reduce invasion. There were no significant effects of pedicularis on either of the Lespedeza species, specifically focusing here on Lespedeza cuneata, which is unfortunate, but it could indicate that pedicularis is really not a good candidate to act as a sort of biocontrol against invasive legumes like Lespedeza cuneata. However, as mentioned before, pedicularis does seem to have potential for aiding in the others instead. And so finally, I would just like to give a shout out to my lab, my advisor, and bee members because I could not have done this work without them, as well as a thank you to Craftsman because they were kind enough to do the organic nitrogen testing using the local soil nitrogen test. And then also a big shout out to Cassie ISU and the site because I could not have done this without your support. Thank you all so much.